This is adaptive and acquired immunity part 2. This is when we can actually, now that we have a foundation in how memory B and T cells are formed, how we can manipulate that immune system in terms of immunization, becoming immunocompetent. So when it comes to active immunity is when an individual receives the immune stimulant or antigen, and that's going to create memory. So this is what we have talked about. We've talked about antigen being recognized by B cells, or antigen being recognized by antigen-presenting cells and activating T cells, and then forming memory. If memory cells are formed, the individual is considered immunocompetent. You are immunocompetent if you have memory cells, which means that you won't get the disease a second time. Now, active immunity is going to take time. It's going to take time for the B cells to recognize antigen, activate and differentiate. It's going to take time for the antigen presenting cells to recognize and then activate and differentiate T cells. And so this takes several days, but the good news is that the effects can last for a very long time, sometimes even for life. Now, active immunity is when you receive antigen. That's how active immunity is acquired. There are two ways that you can receive antigen: naturally, via exposure to the pathogen, or artificially, via a vaccine. There's also something called passive immunity. Passive immunity is not when you receive the antigen, but rather when you receive the immune product or antibodies. So somebody else has seen the antigen, and they have activated and stimulated the humoral immunity, and antibodies have been produced, and you receive those antibodies. Passive immunity is passive because you are protected even though you haven't been exposed. The problem with this is that no memory cells are formed, which means no new antibodies are produced. So no memory cells means that it's going to be temporary, and no new antibodies are produced also means it's temporary. It's temporary because, remember, antibodies are proteins. Proteins are going to eventually break down. They don't last nearly as long as cells do. And so while you have immediate protection, the effectiveness is short. So that's the difference between active immunity and passive immunity. In active immunity, you receive the antigen, and in passive immunity, you receive the antibodies. Now, as with active immunity, there's also natural passive immunity, where you receive antibodies naturally, such as IgG, which crosses the placenta, or IgA in breast milk. You can also receive antibodies artificially. So this little corgi here, please imagine that that is a plasma donation. Because plasma is the liquid, it's the serum that contains all the blood proteins. And what are the blood proteins? A lot of them are antibodies. In fact, when we talk about artificial passive immunity, when we're talking about receiving antibodies artificially, that's also called immunotherapy. Immunotherapy started with using horse serum or horse plasma to prevent tetanus and treat patients that were exposed to diphtheria. I spelled diphtheria wrong there. Now you can take pooled plasma from humans, concentrate those immunoglobulins, purify them, and share them with other humans. So, for example, when the Ebola outbreak happened most recently, a few people actually survived Ebola, and they were one of the greatest hopes for the people who were still experiencing the disease because there is no way to prevent a disease that's already happening. And so if you can help them by boosting their immune system with artificial passive immunity, giving them lots of immunoglobulins to help them clear that infection, um, then that's a great hope for them. The problem with this is going to be a really small pool, such as my example with the people who survived Ebola. It has to be recent, and of course it has to be the same antigen that stimulated those immunoglobulins. And so we can continually expose animals to the antigen in order to create those immunoglobulins, those antibodies, and collect and pool that. But because these are proteins and proteins are antigenic, it is possible that you could take the immunoglobulins from a mouse, say, which was pictured here, and give it to somebody, but it might also cause allergic reactions. Now, artificial active immunity, remember, active immunity is when you receive the antigen. Passive immunity is when you receive the antibodies. So we're talking about antigen again here. This is also vaccines. Vaccines were first attempted with smallpox. This woman, Lady Mary Montagu, she went to the Far East and she brought back smallpox. And what she was doing was rubbing needles in a pustule of people who were infected and then pricking healthy people. And a ton of people died, and this became illegal because this goes absolutely against all the tenets of um, active artificial active immunity, where she was just giving people the infectious agent. Antigen presentation must provide protection, so it must stimulate an immune response and create immunocompetence, create memory cells, but without causing the disease. So this was a huge fail. The next person to attempt a vaccine was Edward Jenner. Edward Jenner observed dairy maids who had been infected with cowpox. Cowpox is very similar to smallpox. It's a pox virus. It's a complex virus. But the dairy maids who had contracted cowpox would only have very mild symptoms. So it's a similar virus, but it's not the same. And when he talked to a milkmaid, she told him that the other milkmaids who had cowpox didn't get smallpox, they were immune. So he focused on this and he prepared material from cowpox, a woman who had been exposed to cowpox or pustule, say, and he used that to inoculate his gardener's son, this small boy. And later, he exposed that boy to smallpox, and the boy was immune. So this was our first successful smallpox vaccine. And a mutated version now today is what we use for smallpox. And in 1973, the CDC declared smallpox completely eradicated. It's only stored in freezers in certain facilities. It's no longer in our population due to vaccines. In fact, smallpox was completely eradicated. There was a spike, and that's because the virus itself mutated. Also, scarlet fever, which is Streptococcus pyogenes, it's a bacterium. That was eradicated due to vaccines. Diphtheria, mortality rate of diphtheria was eradicated due to the development of the diphtheria vaccine. Measles. Measles also, although unfortunately my graph only goes to 2010 and there's been another spike in measles. The spike seen here was when the measles virus mutated, but once the vaccine was redeveloped, mortality dropped to next to nothing due to vaccines. Rubella, measles, mumps, and rubella are common school requirements for American kids to get a vaccine for these. Rubella was also eradicated. Typhoid fever and whooping cough also included on here. So actually, if you just look at mortality of communicable diseases, infectious diseases, after vaccines were first introduced, the death rate died enormously. Now I want to point out Salk vaccine introduced. I want to talk about Dr. Jonas Salk. Dr. Jonas Salk introduced the vaccine for the polio virus. In the 1900s, polio was present. It was endemic, and it wasn't really until the 1950s when the age of polio dropped, and it seemed to be more prevalent in children. And the thing is, polio causes paralysis, and so um, people started to become really quite panicked about this because you would have children who would be paralyzed for life. They would be in braces, as pictured here, and they weren't really sure how this particular virus was being communicated or transmitted, and people were so terrified by this virus that they actually started to believe that maybe it came from ice cream because that's why the young age dropped. And it's not all people, it's an indicator of the panic that was happening at that time. And this outbreak was pandemic. So, pandemic meaning the entire globe was affected by polio. And when you're paralyzed, in these previous pictures I showed legs being paralyzed, and here there's a young person with an arm in a brace that's paralyzed. But one of the things that could become paralyzed is your diaphragm. And so people ended up being put in these contraptions called an iron lung. An iron lung is actually a barometric chamber that breathes for you, because if your diaphragm is paralyzed, you can no longer breathe. And so what this does would increase the pressure to push the air out of your lungs physically, and then decrease the pressure to suck your lungs open, draw your chest up so that you would breathe. And the thing is, when you get put into an iron lung, you're in an iron lung for life. So, while we see all these black and white pictures of the iron lung, some people would spend 70, 80 years in an iron lung. That means that you would have Thanksgiving dinner in an iron lung. You would have your family photos in an iron lung. You would have birthdays and anniversaries in an iron lung. You did all your daily business through the iron lung. You couldn't even pet your own dog because you were stuck in an iron lung. So you can imagine that the stakes were really high for finding some way to not just cure the virus, because once the paralysis happens, it's done, but to prevent it. And so all kinds of money went into researching a cure or a vaccine. In fact, that's where the March of Dimes came from. The March of Dimes was children going around with a little box, begging for money, begging for dimes that they could then put towards researching a cure. Philanthropy was huge. And there was this scientist, Jonas Salk, who had been researching the poliovirus for many years, and he saw this as an opportunity to develop a vaccine, because he was already a bit of an expert. He worked for 7 years and eventually came up with a successful and safe vaccine. And the way he first tested out this vaccine was to inoculate himself, his son, his wife, and everybody in his lab also inoculated themselves, their wives, and their kids. But the vaccine worked, and the world rejoiced. The world rejoiced because now there was a way to prevent this horrible global pandemic that was crippling people and claiming lives and putting them in permanent iron lungs and crutches or dead. And the best part of all of this is that the world knew who Jonas Salk was, Dr. Jonas Salk. And he could have made so much money on this vaccine. He could have just been a quadrillionaire, even in the 50s, right? And he was doing a radio uh, interview, and somebody said to him, well, Dr. Salk, who owns the patent to this? Did you patent your vaccine? And he replied, could you patent the sun? Meaning by that statement that while the sun doesn't belong to any one individual, neither does the polio vaccine. He gave away his polio vaccine to the world without charging a dime for it. So I tell you all of that because what we're going to do is we're going to develop vaccines in our class. We're going to be Dr. Jonas Salk. Now, when you develop a vaccine, you're not going to be Lady Mary Montagu. The purpose is to stimulate the body to create immunocompetence. So artificially provide antigen without causing the disease, but to prime the body so that memory cells are formed so that if there is subsequent exposure to the actual virulent pathogen, the body can respond quickly enough that no disease will result. Now, an ideal vaccine would have all of these qualities. It should not be toxic and have side effects. It should protect against all forms of the pathogen. It should stimulate both humoral and cell-mediated response. It should be long-lasting, lots of memory cells and long-lasting ones. Should not require numerous boosters and doses. And of course, it should be inexpensive, have a long shelf life, and be easy to administer. The problem with any ideal, though, is that the practicality means that we have to give up some of those ideals in order to achieve a true vaccine. There are 4 ways that we can prepare antigen for a vaccine. So we want to prepare an antigen so that when we expose the body to it, we stimulate memory without causing the disease. One way to do that is to kill whole cells or viruses and inactivate them so that they're still antigenic, but are not capable of causing the disease. Another way is to take living cells or active viruses and attenuate them. Attenuation means weakening. So weakening them would mean giving mild side effects, so it would not cause the full-blown disease. You could also use antigenic molecules, which are pieces of the bacterial cells or virus that would stimulate an immune response and memory, but it would not be an actual living organism. Or you could genetically engineer microbes or microbial antigens, and I'm going to talk about each of these 4 and the trade-offs of each of them. So kills whole cells or inactivated viruses. So viruses are not alive, so you can't kill them. There are both vaccines for bacteria and for viruses, and you can do that by killing them or inactivating them. How do we do that? Well, we treat them with formalin, which is like formaldehyde similar, radiation or heat. Anything that's going to kill or inactivate the microbe without changing the antigen structure. It's very important that the antigen still looks like the native virulent form of the organism. So the antigen, or the advantage of this, is that it's going to be safe for most individuals. Age extremes, immunocompromised people, it doesn't matter because this is not a living organism. It is incapable of causing disease. It's going to have a very long shelf life because there's nothing to culture, there's nothing to refrigerate or keep alive. We don't need any safety procedures in case a bottle were to break and somebody were to be exposed to it. It's killed or inactivated. The disadvantage of this type of vaccine preparation is that it's going to require really large doses because the microbe isn't going to replicate within the host or host cells, we need a lot of antigen to introduce to the host so that it creates an equally large group of memory cells. So it's going to require a very large dose and or boosters in order to create a lot of memory. It may also require something called an adjuvant. An adjuvant is something that is going to prolong the retention of the antigen at the site of introduction. Prolong the retention of the antigen at the site of introduction. What that means is sometimes our immune system works so great and is so fast and so efficient that it actually removes the antigen without stimulating as much immune response because the cells are either killed or inactivated, and so you get very little memory response. This also may not activate T cells, so this may be only humoral uh, immune response. So you may only get memory B cells, you may not get memory T cells. So because it is a different type of antigen. And of course, we're not going to get IgA production. And our example of this would be the cholera vaccine and Salk's polio vaccine, Dr. Jonas Salk. Now, our next antigen preparation is live attenuated cells or infectious virus. So attenuated means less virulent or not at all. So they are weakened or they are not at all weakened. And the way that this can be achieved is either by modifying the growth conditions, such as really long-term cultivation. So for example, um, you could you could cultivate microbes so that they really are unable to replicate at 37 degrees, which is body temperature. So you grow them and you select uh, just the cells that can't quite grow at 37, and then you grow them again and you keep selecting until you get these weakened versions. You can also get ones that are unable to replicate in human cells by growing them in non-human culture, or ones that are unable to migrate to their target tissue. So that by introducing them to tissue that is not optimal, so it's not the specific space where that particular pathogen would be able to grow. Modifying the growth conditions or manipulating the genes, yes, you can knock out virulence genes. Let me just rest on that a little bit. So modifying growth conditions is what I talked about here. Long-term cultivation, manipulating the genes is long-term cultivation, right? You're constantly selecting for those that express a certain phenotype. You can also directly manipulate the genes. You can go in and you could knock out a gene for a virulence factor. So for example, uh, an organism that produces a particular toxin. You could go in and think about some of the ways that we can do DNA recombination in in our previous topic. We got conjugation, transformation, transduction, and transposons. Um, or we could even do something called site-directed mutagenesis, where we actually remove or disrupt, mutate a gene so that they can no longer produce a toxin. And so now they are incapable of being virulent, but they are still going to express their same surface antigens. The advantage of this is that these viable organisms are going to be able to multiply in the host, so you don't need as large of doses and you don't need boosters. You're going to have much longer lasting protection because of this, and it will activate both B and T cell immunity. Adjuvants are not going to be required, so all of the disadvantages of the previous are advantages here. And so then you can imagine that the advantages of the previous are going to be the disadvantages here. Because these organisms are are less virulent, and even if they're not virulent, you're still introducing a live organism into an individual. That means that this may not be safe for people who are immunocompromised, immunosuppressed, or pregnant, meaning it could still cause some signs and symptoms. It could still have some effect, and that may not be good for people who are at those age extremes, immunosuppressed, immunocompromised, or pregnant. Um, Another disadvantage is that it may require special storage, right? If this is a living organism, you may need to refrigerate it. You may need to provide it with some sort of preservative so that it doesn't die or degrade. You also may need to have some sort of biohazardous security for something that could be virulent, even if it is less virulent. It may be transmissible. So, for example, we have the um, killed whole cell flu vaccine, which is an injection, and then there is also a nasal flu vaccine, which is live attenuated. You can administer it through the mucous membrane in the nose because it's alive, it can go through, but it is attenuated, so it's weakened and it can still provide protection. When I worked in assisted living, they made everybody, including the residents, but also the staff, get a flu vaccine. And we all had to get the killed whole cell, which was a intramuscular vaccine, because that live attenuated would be too much for the residents. Now, think about it the same way. You go get a live attenuated vaccine and then you go home and kiss your baby or kiss grandma, and you've transmitted that organism to them. It could also mutate back to the wild type phenotype. So remember, wild type phenotype is a non-mutant looking phenotype. So it could mutate to be less virulent, but then it could also back mutate to a virulent phenotype. This is very low chance, but it is possible. And of course, if it requires special storage, it is going to have a shorter shelf life, may require refrigeration, and it may also be limited by antibodies already present in the patient, which means that we may have some immunity already that's going to clear that infection or clear that antigen before we have a chance to form memory. Example would be tuberculosis vaccine was made by subculturing bovine tuberculosis. Subsequent subculturing, and the measles, mumps, and rubella, all three of those were made this way. Acellular or subunit vaccines, I call this the bits and pieces vaccine. So these can be extracted from cultures that are producing these bits and pieces that have been genetically modified to produce them, grow them just for us, like in a chemostat, say. Or you could take a bacterium and you could just shred it up and use those bits and pieces. Or you could use just the antigenic molecule. So you could take a toxin, like an exotoxin, and you could inactivate it and use just that inactivated exotoxin. That's called a toxoid vaccine. Um, For example, the diphtheria and tetanus toxins are toxoid vaccines. Pneumococcal and meningococcal and anthrax vaccines are the kind of shredded bits and pieces ones. Right where I used to live in Lansing, there was a place called Emergent Biosolutions, and it was the only FDA approved place that makes the anthrax vaccine. And the way they do that is they grow an organism, they actually grow it um, in it's a attenuated organism, which means it doesn't actually produce the toxin, which is good because you want double protection. And then they take the attenuated organism and they just shred it up, and they give it to the military. Now, You could also do something like an empty virus that doesn't have the nucleic acids that it needs to replicate, but it does have all the same surface proteins. So that would be like the hepatitis B vaccine, hepatitis B surface proteins. Now, advantages and disadvantages of this one. This is bits and pieces, right? So this is a non-living vaccine. So all the advantages and disadvantages of this vaccine are going to be the same as your other non-living vaccine. Okay. So your killed whole cell or inactivated virus. Genetically engineered microbes and microbial antigens. So now the way that we genetically engineer a vaccine is you start out with the DNA of the antigen. So whatever that molecule is, you have the gene that makes that molecule, and you clone it into a vector. So we call that vector a plasmid here. So we've got the DNA that codes for the surface antigen of this virus cloned into a plasmid. And then we can transform that plasmid into, say, a yeast cell or a bacterial cell that is benign, that is not going to be virulent. And now that yeast cell can, you can either extract that surface protein or, and give that as a bits and pieces molecule, or you can inject the entire yeast cell into your patient, your subject, and now, and I say inject, you can introduce the yeast cell. Not all vaccines are shots. But you can introduce that yeast cell into your subject, and now the yeast cell is avirulent. It cannot cause a disease, but it is still expressing the surface antigen protein so that the body will recognize that, and then if it ever encountered the real virus later, it would already have the memory cells to respond to that. Um, this is what we call a Trojan horse vaccine because this is a benign organism that is expressing that virulent organism's surface proteins. Um, and so you can do that by a Trojan horse, or you can do it, like I said, by extracting them. So that's a genetically engineered one. You don't have to use yeast. You can use other viruses. You can use tissue cultures or egg embryos. All of these are ways that you can get those recombinant vaccines. This particular version of a genetically engineered vaccine is a recombinant vaccine. An example would be the Lyme disease vaccine, hepatitis V, C, and herpes simplex virus vaccines. Now we also have what we call a DNA vaccine. So we've taken the DNA of the antigen and cloned it into the vector. So we've got the DNA of the pathogen cloned into the plasmid. Now we have the plasmid with that DNA, and we can introduce that plasmid directly into the subject. That's why this is called a DNA vaccine. And so right into the subject, and our cells then would take up that plasmid. We would incorporate it into our chromosome and start expressing those uh, proteins that the DNA from the pathogen coded for. Um, Now our host cells are expressing non-host proteins, and so our own host cells start to look like the pathogen. Our own body can degrade and clear those away and create a memory response, and never actually experience the disease. Examples of this were the original smallpox vaccine and the adenovirus. Now, these are living vaccines. At least the recombinant vaccine is going to be living. So all the advantages and disadvantages of our other living vaccine preparation are going to apply here. This one is um, a little bit more like, say, a bits and pieces vaccine because it's our own cells. But you can kind of piece together what the advantages would be. These cells can replicate depending on how much plasmid has been introduced. And yet it's not going to require special storage because it's not really alive. So this one kind of takes a little bit of each. I mentioned that that vaccines are not always shots. Students tend to always associate vaccines with viruses and shots, and that is not true. You can have shots, you can have sub-Q, intramuscular, intradermal, but you can also have oral vaccines, intranasal vaccines. Um, and there are vaccines for bacteria. Um, Tdap, for example, tetanus, diphtheria, right? The pertussis, those are all bacterial vaccines. Adjuvant. Remember, adjuvant prolongs the retention of the antigen at the site of introduction so that the host cells can have more contact. So that adjuvants tend to have a bad reputation, but that's their purpose. When it comes to real vaccines, many years of trials in animals have to occur first, cell culture, then animals, then humans. But in general, vaccines are very safe. That's the whole point of them, is to prevent disease. Recalls and replacements do happen, and it's usually to reduce side effects. They're always trying to make the side effects less and less, and it's usually not due to the antigen itself, but rather all the other things that go into it, like preservatives or antigens that might cause allergic reactions, or maybe they can run it through something other than, say, eggs, who are people who are allergic to eggs, or yeast. The risk of the infection should always outweigh the risk of vaccination, always, and that's why. And then lastly, vaccines do not cause diabetes, asthma, or autism. This is really, really important because in our recent history, there was a research scientist named Wakefield. And Wakefield, his name is Mud. And the reason his name is Mud is because what he did was he published a bunch of research that correlated vaccines with these other diseases. Now, in science, we have a phrase called correlation does not equal causation, meaning just because there's a high incidence of something doesn't mean that it actually caused it to happen. So, for example, um, you may be driving along and you may notice that the police have pulled over red cars for speeding. It just seems to be a really high correlation between the number of cars that got pulled over for speeding are red. But the police aren't pulling over the red cars because they're red, right? Correlation did not equal causation. The police pulled over the red cars because they were speeding, right? So that's really, really important. And in fact, Wakefield, after he published this stuff, he retracted it, and then all the publications retracted it, and the scientific community has shunned him. That's what I mean by his name is Mud. But the problem is that the damage has been done. With the advent of social media and the ability for people who have absolutely no scientific education to be able to stand and spout information as if it is fact without any fact checking, without any way to have a peer response, a peer reviewed scientific response. These things still seem to be radiating through our culture. People who don't understand that just because an adjuvant has something like aluminum in it doesn't mean it's poison. Okay, so off of my point here and onto something else. Vaccines are incredibly important. Vaccines prevent disease. They prevent disease in you and me. Um, and not just this lifelong immunocompetence, but they also can prevent disease in people around us via something called herd immunity. Herd immunity is when you have enough people who are immunocompetent, whether by vaccines or by natural active immunity. Remember what natural active immunity was? Active immunity is exposure to antigen. So natural is when you get sick and get better, right? When you have enough individuals that are immunocompetent, they can actually interrupt transmission. So, for example, Perhaps you have a plane full of people, and one person in the back of the plane um, is immunocompromised or not immunocompetent. I shouldn't use those two words together, I'm sorry. Someone who's susceptible. And then you have a plane full of people who are immunocompetent. And then you have one person way up at the front of the plane who is infected. All of those people that are immunocompetent are not going to be able to contract that infectious agent. They're not going to be able to pass it along. Now, obviously, infection doesn't pass that quickly. That's just an example. Here's a picture version of herd immunity. So herd immunity is when a majority of population, so you actually have to have a threshold number of individuals to interrupt transmission. It can't just be a few, such as the middle picture, because transmission can still happen. It has to be the majority of the population. It eliminates the population as a reservoir for the pathogen. So the advantage of herd immunity is to protect people who are immunodeficient. That's the word I was supposed to use. Or people who are not able to receive a vaccine or develop active immunity. And the disadvantage is you have to maintain that threshold number. It's very important to keep the immunocompetence level high. And there are two ways that herd immunity are developed. Right? I just mentioned them, right? Vaccines and getting sick and getting better. Remember, active immunity is exposure to antigen. Passive immunity, you receive antibodies. So, immunoglobulins transferred from mother to baby is an example of, pause your slides, natural passive immunity. Yes. Passive immunity, you receive immunoglobulins. Immunoglobulins transferred in one person's plasma to another is an example of artificial passive, right? Passive immunity, you receive immunoglobulins. You receive antibodies in passive immunity. What is that also called? Artificial passive immunity? Immunotherapy. Immunoglobulins formed after exposure to the flu virus is an example of, pause your slide, natural active immunity. Active immunity, you receive antigen. Active immunity, you receive antigen. Okay, NCLEX style question. Sorry about the spacing here. The major histocompatibility complex is a gene complex that gives rise to a series of glycoproteins, MHC molecules, found on all cells except, so MHC is on all cells except, pause your slide, red blood cells, right? MHC1 is on all nucleated cells, and red blood cells don't have a nucleus. Remember, MHC1, I'm number 1. It's on all self cells. MHC2 is only on our antigen-presenting cells. Which characteristic is associated with passive immunity? Pause your slide. Immediate protection. Passive immunity is short term because you receive antibodies. So you have the product of active immunity. You receive antibodies, you receive immunoglobulins. You're immediately protected, but it's very short term. And the reason that it's short term is because there are no memory cells, no new antibodies are produced, and antibodies are protein and they break down. That is the end of adaptive and acquired immunity. I know it was a long one, but I wanted to get through it. All right, stopping the slides now.